Welcome everyone back to our class. Oh, you are really study hard. Ever wondered what tiny protein is like a bouncer at your body's club door, kicking out bacteria and sometimes causing a ruckus? Meet LL-37. It's not just any protein, it's the human cathelicidin antimicrobial peptide, born from the CAMP gene on chromosome 3p21.3. Its story starts in 1995 when scientists first caught sight of its precursor, then confirmed its power in 1996, and finally isolated it in action in 1998. Pretty cool origin story for such a tiny powerhouse. So where does this LL-37 superhero hang out? It's got a VIP list of cells. Think of it as living in both the city's defense force and the neighborhoods themselves. You'll find it chilling in immune cells like neutrophils, those granule-packed warriors, monocytes, mast cells, and T cells. But it's also found in epithelial cells, the skin's keratinocytes, the lining of our lungs and gut, even the mouth. High concentrations? Bone marrow, where myeloid cells mature, and tissues like skin, oral mucosa, lungs, and intestines. It even tags along with lactoferrin in neutrophil granules and shows up at inflammatory hotspots like psoriasis plaques or inflamed bowel tissue. Basically, wherever there's a fight brewing, LL-37 is likely there too. Alright, let's talk about LL-37's superpowers. Its main gig is innate immunity basically. It's the first responder against bacterial invaders. It punches holes in bacterial membranes, doesn't care if they're gram-positive or negative, and neutralizes nasty endotoxins like LPS. But here's the twist. It's a bit of a Janus figure, playing both sides. On one hand, it's pro-inflammatory, calling in immune cell reinforcements, neutrophils, monocytes, T cells, and activating inflammasomes via P2X7 receptors to release IL-1 beta. On the other hand, it can be anti-inflammatory, blocking LPS from binding to TLR4, thus calming down the inflammation. And it's not done yet. It helps repair damaged epithelial tissues, promotes new blood vessel growth, induces netosis, those web-like traps from neutrophils, and even fights viruses directly. Talk about versatile. So how does LL-37 actually pull these levers? It's all about the receptors it interacts with. Think of it as having different keys for different locks. The P2X7 receptor is a big one. Binding it triggers ATP release, activates caspase 1, and leads to mature IL-1 beta, a key inflammatory signal. Then there's FPRL1, which is crucial for attracting immune cells to the scene and mobilizing calcium. The toll-like receptors, or TLRs, add another layer. TLR4 often dampens inflammation, while TLR9 gets activated by complexes involving LL-37, pushing the inflammatory pedal down. And don't forget the MAPK pathway, where ERK and JNK activation leads to COX-2 and PGE2 production. Each receptor interaction sparks a cascade of effects, like releasing IL-1 beta through P2X7, guiding cells via FPRL1, or boosting epithelial growth through EP3. It's a complex network, but incredibly effective. Now, let's bring this down to earth. How does LL-37 play out in real life? Well, it's deeply involved in diseases. In psoriasis, it's overexpressed, driving keratinocyte proliferation and that interferon storm. In IBD, it ramps up T-cell inflammation in the gut. In SLE, it stabilizes immune complexes and boosts interferon production. But it's not all doom and gloom. This duality makes it a fascinating therapeutic target. We could use agonists to boost it in sepsis or against viruses, or inhibitors to calm it down in autoimmune diseases. Scientists are even tweaking it, like creating the KR12 peptide, which keeps its antibacterial punch while reducing toxicity. The big takeaway? LL-37 is a double-edged sword. Understanding its context, its concentration, its environment is absolutely crucial for harnessing its power safely. That's the magic and the challenge of this incredible peptide.